Experiment 3. Determination of the focal length of a concave mirror and a convex lens. A. To determine the focal length of A, that is, a concave mirror, B. Convex lens, by obtaining the image of a distant object. Requirements: a concave mirror, a convex lens, a meter scale, a white screen, a mirror holder, a lens stand, and a screen holder. Procedure. Now first we will mount a concave mirror in the holder. Select a distant object like an electric pole. Turn the reflecting surface of the mirror towards the electric pole. Now mount a white screen in the screen holder and place it in front of the mirror. Number 4. Move the screen towards or away from the mirror and obtain a sharp image of the electric pole. Measure the distance between the pole P of the mirror and the screen, that is the image. Now repeat the procedure by selecting 2 more distant objects like a tree or a house, a telephone pole. Repeat the procedure from 1 to 6 by using a convex lens. In this case, measure the distance between the optical center O of the lens and the screen. Observation table. Table for the concave mirror. Now, the columns are of serial number. We can write the distant object over here and distance between the pole of the mirror and image in centimeter over here. And we will find the mean distance. Next is table for convex lens. Serial number, distant objects, we can write them here. Distance between the optical center of the lens and image in centimeter. And we can find the mean distance. Inference. Focal length of concave mirror is equal to mean distance, and that is 9.7 centimeter. Number 2. Focal length of the convex lens, which is equal to mean distance, that is 18.7 centimeter. Experiment 4. Measuring the angle of incidence, the angle of refraction, the angle of emergence, and interpreting the results. A. To trace the path of ray of light passing through a rectangular glass slab for different angles of incidence, and to measure the angle of incidence, the angle of refraction, the angle of emergence, and interpret the results. Requirements: a rectangular glass slab, a drawing board, a drawing paper, drawing pins, pinch protractor. Procedure: fix the drawing paper on the drawing board using drawing pins. Place the rectangular glass slab on it and mark its boundary with a sharp pencil. Remove the glass slab and draw normal M1, N1 at the point O. Draw a line AO which forms an angle of incidence I of 30 degrees with normal M1, N1. Erect pins P1 and P2 on ray AO such that the pins are vertical and well separated. Place the glass slab properly in its original position. Observe the images of the bases of pin P1 and P2 through the glass slab from the opposite side and fix two pins P3 and P4 such that the bases of these pins are exactly in line with the images of bases of pins P1 and P2. Remove pins P3 and P4 and mark their positions by small circles. Draw a line through points P3 and P4. Remove pins P1 and P2 and the glass slab. Join OB and draw normal M2, N2 at point B. Measure the angles I, R and E. Repeat the above procedure for two more angles of incidence of 45 degrees and 60 degrees. Now here is the observation table. With columns serial number, angles of incidence I, angle of refraction R, angle of emergence E. Inference. The path of the ray of angle of incidence of 30 degrees is AOBC. Incident ray AO and emergent ray BC are parallel to each other. The lateral shift is equal to CD. This shift depends upon the thickness T of the glass slab, the refractive index of glass, and the angle of incidence. The angle of incidence I is equal to angle of emergence E. Experiment 5. Study of the dependence of current on the potential difference across a resistor and determination of its resistance. A. To study the dependence of current I on the potential difference V across a resistor and determine its resistance. Also plot a graph between V and current I to verify Ohm's law. Requirement: a resistor, nichrome wire or manganese wire, a rheostat, dry cells or a battery eliminator, a plug key, a voltmeter, and a meter, connecting wires and a sandpaper. Procedure: select a voltmeter and a meter of proper range and note their zero error and least count. Connect the circuit as shown in the figure with connecting wires. Keep the plug key open. Close the circuit by inserting the plug in the key. Adjust the rheostat by sliding the variable terminal till desirable reading is obtained in the ammeter. Note the reading of the voltmeter and open the circuit by removing the plug from the key. In place of a single cell, connect two cells in the series in the circuit to increase the potential difference. Close the plug key to complete the circuit. Note the reading shown by the ammeter and the voltmeter. Take 3 more readings by increasing the number of cells from 2 to 4. Keep the plug key open after taking all readings and disconnect the circuit. Plot a graph between V and I. Observations. Number 1. Least count of the ammeter is. Least count of the voltmeter is. Zero error of the meter is. Zero error of the voltmeter is. Now let us see the observation table. Mean value of resistance is. Results. Current I in the circuit increases or decreases with increase or decrease in voltage V. That is, V upon I is equal to constant, or I is directly proportionate to V. The value of unknown resistance is ohms. The graph of I versus V is a straight line passing through the origin of 0, 0. This is the graph of potential difference and current. Note, zero error. When there is no current in the circuit, the pointer in an ammeter as well as in a voltmeter should be at the 0 mark. If suppose the pointer is at or on some scale division x instead of 0, then x should be subtracted from the observed readings to get the corrected reading. Experiment number 6. Determination of the equivalent resistance of two resistors connected in series. Aim. To determine the equivalent resistance of two resistors connected in series. Requirements: 2 unknown resistors, a rheostat, a voltmeter, an ammeter, a plug key, dry cell, or a battery eliminator connecting wires, a sandpaper. Let us first draw circuit diagram. Procedure. Select the voltmeter and the ammeter of suitable range. Note the least count and zero error of the voltmeter and the ammeter. Connect the electrical circuit as shown in the circuit diagram. Show the connections to the teacher or examiner. In the first step, connect the voltmeter across resistor R1. Put the plug in the key and adjust the rheostat to obtain the desired current in the ammeter. Note the readings of the voltmeter and the ammeter. Obtain 4 steps of reading for different currents. Then connect the voltmeter across resistor R2 and again obtain 4 steps of reading as above. Finally, connect the voltmeter across both the resistors. Here also obtain 4 sets of readings as above. Remove the plug from the key and disconnect the circuit. Observation number 1. Least count of the voltmeter is 0 volts. Number 2. Least count of the ammeter is 0 amperes. Number 3. Zero error of the voltmeter is 0 volts. Number 4. Zero error of the ammeter is 0 amperes. Now let us fill the observation table. It has columns, step, set of observations, ammeter reading I, which is observed and corrected, voltmeter reading V, observed, corrected, V upon I, ohms. For R1, for first set of observation, ammeter reading observed is 0.15, ammeter reading corrected is 0.15, voltmeter reading observed is 0.45, voltmeter reading corrected is 0.45, so it is 3.0 ohms. For second set of observations, Ammeter reading observed is 0.20. Ammeter reading corrected is 0.20. Voltmeter reading observed is 0.60. Voltmeter reading corrected is 0.60. It is 3.0 ohms. For R1, here we have third set of observations. Ammeter reading observed 0.26. Ammeter reading corrected 0.26. Voltmeter reading observed 0.80. Voltmeter reading corrected 0.80. It is 3.02 ohms. For R2 set of observations, for first set of observation, ammeter reading observed 0.30. Ammeter reading corrected 0.30. Voltmeter reading observed 0.90. Voltmeter reading corrected 0.90. It is 3.0 ohms. For second set of observations, ammeter reading observed 0.35. Ammeter reading corrected 0.35. Voltmeter reading observed 1.10. Voltmeter reading corrected 1.10. It is 3.1 ohms. For third set of observations, ammeter reading observed 0.40. Ammeter reading corrected 0.40. Voltmeter reading observed 1.20. Voltmeter reading corrected 1.20. It is 3.0 ohms. For Rs is equal to R1 and R2 connected together in series. For first set of observations, ammeter reading observed 0.28. Ammeter reading corrected 0.28. Voltmeter reading observed 1.68. Voltmeter reading corrected 1.68. It is 6 ohms. For second set of observations, ammeter reading observed 0.26. Ammeter reading corrected 0.26. Voltmeter reading observed 1.58. Voltmeter reading corrected 1.58. It is 6.07 ohms. For third set of observations, ammeter reading observed 0.27. Ammeter reading corrected 0.27. Voltmeter reading observed 1.62. Voltmeter reading corrected 1.62. It is 6.00 ohms. Now let us see the calculations. Number 1. Mean resistance R1 is 3 plus 3 plus 3.02 upon 3. That is 9.02 upon 3, which is 3.00 ohms. Number 2. Mean resistance R2 is 3 plus 3.1 plus 3 upon 3. That is 9.1 upon 3, which is 3.03 ohms. Number 3. Total resistance Rs is equal to R1 plus R2, that is 6.03 ohms. Number 4. Mean equivalent resistance Rs when R1 and R2 connected together in series is equal to 6 plus 6.07 plus 6 upon 3, that is 18.07 upon 3, which is 6.02 ohms. The result is number 1. The equivalent resistance of two resistors in series is equal to 6.02 ohms. Number 2. The relation Rs is equal to R1 plus R2 is experimentally verified. Experiment number 7. Determination of the equivalent resistance of two resistors connected in parallel. Aim. To determine the equivalent resistance of two resistors connected in parallel. Requirements. 2 unknown resistors, a rheostat, a voltmeter, an ammeter, a plug key, a battery, connecting wires, a sandpaper. Let us first draw circuit diagram. Procedure. Select the voltmeter and the ammeter of suitable range. Note the least count and zero error of the voltmeter and the ammeter. Connect the electrical circuit as shown in the circuit diagram. Show the connections to the teacher or examiner. In the first step, connect the ammeter in series with resistor R1. Put the plug in the key and adjust the rheostat to obtain the desired current in the ammeter. Note the reading of the voltmeter. Obtain 4 sets of such readings for different currents. Then connect the ammeter in series with resistor R2 and again obtain 4 sets of readings as above. Finally, connect the ammeter in the main line across both resistors. Here also obtain 4 sets of readings as above. Remove the plug from the key and disconnect the circuit. Observation. Number 1. Least count of the voltmeter is 0 volts. Number 2. Least count of the ammeter is 0 amperes. Number 3. Zero error of the voltmeter is 0 volts. Number 4. Zero error of the ammeter is 0 ampere. Now let us fill the observation table with column step, set of observations, ammeter reading I, observed, corrected, voltmeter V, observed, corrected, V upon I, ohms. For R1, for first set of observations, ammeter reading observed 0.15, ammeter reading corrected 0.15, voltmeter reading observed 0.10, voltmeter reading corrected 0.10, it is 0.66 ohms. For second set of observations, ammeter reading observed 0.30, ammeter reading corrected 0.30, voltmeter observed 0.20, voltmeter corrected 0.20, it is 0.66 ohms. For third set of observations, ammeter reading observed 0.45, ammeter reading corrected 0.45, voltmeter observed 0.30, voltmeter corrected 0.30, it is 0.66 ohms. For R2, for first set of observations, ammeter reading observed 0.60, ammeter reading corrected 0.60, voltmeter reading observed 0.40, voltmeter reading corrected 0.40, it is 0.66 ohms. For second set of observations, ammeter reading observed 0.75, ammeter reading corrected 0.75, voltmeter reading observed 0.50, voltmeter reading corrected 0.50, and it is 0.66 ohms. For the third set of observations, ammeter reading observed 0.90, ammeter reading corrected 0.90, voltmeter reading observed 0.60, voltmeter reading corrected 0.60, it is 0.66 ohms. For RP, when R1 and R2 connected together parallel, first set of observations, ammeter reading observed 0.75, ammeter reading corrected 0.75, voltmeter reading observed 0.24, voltmeter reading corrected 0.24, it is 0.33 volts. For second set of observations, ammeter reading observed 1.05, ammeter reading corrected 1.05, voltmeter reading observed 0.34, voltmeter reading corrected 0.34, it is 0.32 ohms. For third set of observations, ammeter reading observed 1.35, ammeter reading corrected 1.35, voltmeter reading observed 0.45, Voltmeter reading corrected 0.45. It is 0.33 ohms. Calculations. Number 1. Mean resistance R1 is equal to 0.66 plus 0.66 plus 0.66 upon 3, that is 0.66 ohms. Number 2. Mean resistance R2 is equal to 0.66 plus 0.66 plus 0.66 upon 3, that is 0.66 ohms. Number 3. Total resistance 1 upon Rp is equal to 1 upon R1 plus 1 upon R2, which is equal to 1 upon 0.66 plus 1 upon 0.66. So 1 upon Rp is equal to 0.66 plus 0.66 upon 0.66 into 0.66. It is equal to 1.32 upon 0.4356. 1 upon Rp is equal to 3.03. So Rp is equal to 1 upon 3.03, that is 0.33 ohms. Number 4. Mean equivalent resistance Rp when R1 and R2 connected together in parallel is equal to 0.33 ohms. Result. Number 1. The equivalent resistance of two resistors in parallel is equal to 0.33 ohms. Number 2. The relation 1 upon Rp is equal to 1 upon R1 plus 1 upon R2 is experimentally verified. Experiment 9. Experiment 8. Study of stomata. A. To prepare a temporary mount of leaf peel to show its stomata. Requirements: any dicot leaf, glass slides, microscope, distilled water, dropper, glycerine, cover slips, forceps, etc. Procedure: take a leaf and observe its parts. In case of a small leaf, place it on a glass slide and put a drop of glycerin on it. Cover it with cover slip. Observe this temporary mount of leaf under a microscope. For observing stomata, select a plant and keep it exposed to sunlight for a few hours. Take a leaf of such plant, tear it tangentially from its lower surface to obtain a thin peel. Mount this thin peel on a glass slide, add a few drops of water on it and expose it to sunlight. Observe the leaf peel under microscope. Note down your observations. Keep this slide in darkness for 10 to 15 minutes and again observe it under a microscope. Note down your observations. Observations. The stomata gets widely opened after exposing to sunlight. Oblong or oval pores can be seen between two kidney-shaped guard cells. Number 2. After keeping the mount in darkness, the stoma is found to be closed because guard cells lose turgidity in darkness. Experiment 9. To prove that light is necessary for photosynthesis. A. To show experimentally that light is necessary for photosynthesis. Requirements: a potted plant, iodine solution, black paper strip, paper clips, water bath. Procedure number 1. Select a potted plant and keep it in complete darkness for about 48 hours. The plant will be destarched as photosynthesis is not taking place due to absence of sunlight. With the help of clips, fix a strip of black paper on both the sides of a single leaf of it. Keep the potted plant in sunlight for about 8 hours so that the process of photosynthesis takes place. Remove the black strip and boil the leaf in alcohol or a water bath so that chlorophyll is removed. Wash the leaf with water and put a few drops of iodine on it. Observe the leaf. Observations. The region of the leaf which was exposed to sunlight turns blue in color. The region of the leaf covered with black strip turns brownish in color. Inference. This experiment shows that sunlight is necessary for photosynthesis. Experiment 10. To prove that carbon dioxide is given out during respiration. Aim. To show experimentally that carbon dioxide is given out during respiration. Requirements: a conical flask, beaker, test tube, KOH pellets, a tube bent at right angles, cork, thread, soaked gram seeds, colored water, etc. Procedure: soak gram seeds overnight in water and germinate them by placing the seeds in moist cotton wool for 1 to 2 days. Place moist germinated seeds in conical flask so that respiration may continue for longer time. Suspend a test tube with KOH pellets in the conical flask. Fit the cork with bent glass tube to the conical flask. Keep the connection airtight. Dip the free end of the bent glass tube in a beaker containing colored water. Mark the initial level of water in the bent tube. Keep this setup undisturbed for about 15 minutes and mark the final level of water in the bent tube. Note down the observations. Observations. CO2 evolved during respiration of germinating seeds will be absorbed by KOH pellets. As KOH is hygroscopic in nature, it absorbs CO2 which is liberated during the respiration of gram seeds. This creates a partial vacuum in the conical flask. So, due to the lower air pressure in flask, the water level in the bent tube rises. Inference: carbon dioxide is given out during respiration in flasks. Experiment 11. Study of binary fission in amoeba and budding in hydra. Aim: to study A. Binary fission in amoeba, B. Budding in hydra with the help of permanent slides or charts. Requirements: charts of binary fission in amoeba and budding in hydra. Procedure number 1. Observe the charts showing binary fission in amoeba and budding in hydra. Number 2. Note down the observations. Observations. A. Binary fission in amoeba. Binary fission starts with the division of nucleus. The cytoplasm shows the formation of a transverse furrow after the division of nucleus. This furrow deepens and divides the amoeba into two daughter amoebae. B. Budding in hydra. Hydra produces a protuberance on the lateral side of the body wall. The protuberance later develops into young hydra by sharing body wall from parent hydra. The bud increases in size and volume by the division of cells. After its development is complete, the bud detaches from parent body and starts an independent life. Experiment 12. Determination of the percentage of water absorbed by raisins. A. To determine the percentage of water absorbed by raisins. Requirement: measuring cylinders, raisins, water, beaker, petri dish, thermometer, equiarm balance, etc. Procedure: take a measuring cylinder and take out 20 grams of raisin in it. Add about 50 milliliter water to it. Measure the temperature of the setup in degrees Celsius and convert it into Kelvin. Let the setup remain undisturbed for up to 2 hours. Then take out the raisins and spread them on a filter paper so that excess of water is absorbed. Make a note of final reduced volume of water. Weigh the raisin and note down the final weight. Repeat the experiment to calculate the percentage of water absorbed. Observations. Number 1. After 2 hours, raisins will swell. Number 2. When the swollen raisins are taken out for weighing, the volume of water in measuring cylinder is found to be reduced. Calculations. Initial volume of water X is 50 milliliter. Initial weight of raisins A 20 grams. Final weight of raisins B 30 grams. Final reduced volume of water Y 37.5 milliliter. Increase in weight of raisins B minus A is 10 grams. Water absorbed is equal to increase in weight, that is 10 grams. The percentage of water absorbed by raisin is B minus A upon A into 100. Temperature is equal to T degrees Celsius, that is 35 degrees Celsius. It is equal to 273 plus 35 degrees Celsius, that is 308 degree Kelvin. Inference: the percentage of water absorbed by raisins at temperature 308 Kelvin is 50%.